All right, everyone, we're going to go ahead and get started. Thank you so much for being here. For those folks in the room, thank you for braving the rain. It is pretty torrential out there, so thanks for coming out. We appreciate you being here. My name is Cinnamon. I'm the Assistant Director of Research Support here at Hatfield, and I will be your host for today. As all of you in this room, I can look around and see, no, this is a hybrid event. So if folks online and folks in the room, for folks that are online, your mics, cameras, and screen shares have been disabled, but please use your chat to put any questions you might have into the question box. And our volunteer, Roseanne, will make sure that we either address those questions or we get those questions to our speaker for today. For folks in the room, you know this already, but please raise your hand when we get to the question and answer, and I'll bring you a mic so that folks online can hear those questions and they can be engaged in this process as well. And as a reminder, if you are a student taking this for credit, make sure your name gets on that special sign-in sheet that says for students. That is one way we track that you were actually here, and so you can get credit for being in the room today. So again, thanks everyone for being here. I'm going to move this along because I'm so excited to hear our speaker today. But I wanted to make one quick announcement for next week's seminar, which is already November 7th. I don't know when that happened. But Marley Parker is a freelance science communicator and collaborator working with Bill Chadwick, and she is going to give our seminar next week at 3:30 about producing compelling stories to promote ocean research. And so that's a slightly different topic for us, but I think it's going to be pretty amazing. So I hope you're able to come back and join us next week. But for today, we have Julia Parrish here, and we talked last night. I'm going to do it again today, and we are just going to get her started. So she's going to introduce herself to all of you as I hand this off to her. Are you ready?
Am now.
Perfect.
Okay, great. All right. Hi, everyone. Recovering from a cold, so I sound stranger than normal. Sorry. So thanks for coming out on a rainy Thursday. My name's Julia Parrish, and I'm a professor at the University of Washington in Seattle, where I've been for, gosh, a heck of a long time. And I have a professorship in the School of Aquatic and Fishery Sciences, and also in the biology department in the College of Arts and Sciences. And my day job is that I'm the Associate Dean for Academics of our College of the Environment there. So a lot of time working with faculty and students and postdocs to develop new teaching learning opportunities there, which is really fun to be that involved in formal education, including at our marine lab, which is a tiny little place compared to this lovely lab, the Friday Harbor Labs. So nights and weekends, the real passion of my life has been what used to be a part and now is all of my lab, the Coastal Observation and Seabird Survey Team, or COAST for short. And that's the majority of the work that I'm going to talk to you about today. And just to say that most of the, hold on, let's try this. There. Most of the work that I'm going to talk about today is a collaboration with Tim Jones, who's now with the British Antarctic Survey, but was a postdoc and then an acting assistant professor with COAST in Seattle for many years. And let me say that all of the work that I'm presenting is work of many, many people, including folks here at Hatfield, but really resting on the quantitative prowess of Tim. Okay, so I want to think about physical timescales provoking ecosystem response in marine systems, and really think about marine birds, so seabirds, but also marine waterfowl and shorebirds, and acknowledge that they are feeding at two trophic levels there at the zooplankton level, so big copepods and amphipods and isopods and small krill, and also at the forage fish level. So those small silvery schooling things that spend their life in groups like herring or anchovies, but also first years of very much larger and even commercially important fish, including salmon and rockfish and pollock. And so in thinking about how physical timescales have an effect, when we think about the effect seasonally, there's a lot of ecosystem forcing that happens. So just from one year to the next, change in seasonal forcing can really rocket up to the top of the food web and have an effect on things like seabirds or marine mammals or large fishes. But I also want to get you to think about something that's happening on between years and decade scales that might cause more long-term shifts in an ecosystem. And although at any one point in time, we can always say, wow, we're in a new time, like this is different than it ever has been before. Arguably true, at least in some scales. We're always thinking in the short term within year. So I want you to like stretch your mind in between what's happening year to year and what's happening decade to decade. And I also want to just alert you that within the Northeast Pacific, there are arguably about 150 million seabirds of many, many different species. So it's kind of hard to get a pulse from year to year on what's going on because changes in energy may be absorbed by one part or another part of that whole big community of seabirds. And before I actually talk about coast, I just want to give a nod to early work that I did when I was a starving postdoc on this, my favorite bird, the common murre, shown here in its football with wings configuration. I talked about this last night, and that's how I describe all of the alcids, and that's because they shrink their neck in and they have short tails, so they come to a point at both ends. So they're not very good at flying in air, but they have done that delicate trade-off between flying in air and flying underwater. So these guys can, at sort of half mass of wings outstretched, fly underwater and to several football fields in depth. So basically there is no place, for instance, that an adult salmon would forage on the shelf that you also won't find this bird foraging. And they're eating pretty much the same things. Well, I did a lot of work on murre colonies, asking questions about what makes populations of these guys go up and down over time. And I was pretty concentrating on predators, including predators on the adult murres themselves, and also disturbers. This is a disturber, not a predator. The adults of this species, adult bald eagles, are pretty good predators. In fact, in some places where eagles and murres co-occur that are on offshore islands, for instance, Triangle Island, which is at the northwest tip of Vancouver Island. So Triangle Island is the 5th island out in the Scott Island archipelago. So it's way the heck out there, right on the edge of the shelf. And there are 5 eagle eyries there, and there also is a fair-sized colony of murres. And the eagles that live there have divided the island territory up into different feeding territories. If you know anything about eagles, they maintain feeding territories separate from where they're breeding. And there's one eyrie that's overlapping the murres, and the pair that nests in that nest, they can get a murre in about 35 seconds. They are very, very, very good predators. But juvenile eagles are very bad predators. In fact, the largest thing that kills juvenile eagles is ineptitude, because they can starve in the midst of plenty. They need to learn what prey is, and they start off by stealing. Okay. And so when they're on murre colonies, and this is a picture of Yaquina from a few decades ago, when they're on murre colonies, they do a lot of disturbing, a lot of flying around. The juvenile that's landed here is surrounded, you may see, by little dots. All those dots are murre eggs. There are hundreds and hundreds of them. And you can see that the murres have faded back away from the eagle. Good thing, right? And you can see that the gulls are on their way in. So this eagle won't get any murres, but it will keep the murres off their eggs. And that allows the gulls to come in and take the eggs. And so there's this really interesting cycle between gulls and eagles and murres, depending on how many of them there are in the situation that is not so good for the murres. And I spent maybe the first 6 or 7 years of my seabird life looking at that interaction. And more or less, it looks like this. So within that interior triangle, eagles have a negative or red impact on the murres directly through predation. They also facilitate gulls. So eagles actually have a positive and a negative impact on gulls. Eagles will eat gulls, but they also facilitate the the gulls going in and really depredating the eggs from murres. So you can see that there's 2 red arrows from gulls and eagles to murres. That was most of the ecology work that I did. And some of the larger scale work when we were adding humans into the mix was really asking the question, well, have humans changed this tripartite interaction between these 3 species? Because these 3 species co-occur all the way up and down the West Coast and certainly throughout Alaska. So it's not that there's one that's introduced, for instance. This is a normal long-term interaction. However, you can see that humans have actually a pretty negative impact on murres, at least that we were looking at the time, not least of which is oil spills. And whenever there's an oil spill on the Pacific coast, murres are arguably the primary species that are killed, at least that float in dead. Fisheries also have a negative impact on murres through bycatch. So coastal fisheries, certainly including gillnet gear, have a negative impact on murres. So negative impacts from humans on murres, but also positive impacts on eagles and on gulls. Gulls, because we provide gulls with all sorts of additional feeding opportunities and also breeding opportunities. And a positive impact on eagles because we have made a concerted effort to conserve and improve the population of eagles, and we've been successful in doing that. So when you add humans to this ecological interaction, you can sort of push the negative impact on murres. And so even back in the 90s, I was asking myself, will common murres become uncommon? Because at least in the main colony that I worked on, which is not Yaquina, I had the privilege of working there and setting up some reproductive success plots there in the 90s. Tatoosh Island, where I work, which is at the northwest tip of Washington state, the murres were in decline and a pretty steady decline as a function of all of these interactions. So I wasn't really thinking about climate and climate change at the time because nobody was, unless they were atmospheric scientists, and nobody listened to those guys anyway at the time. But I do have to say that even then, thinking about what was going on and looking at what was going on, one of the things that we were interested in was what are the murres eating? And the answer is lots and lots and lots of different things, at least on Tatoosh. So they have a pretty broad diet and it changes from year to year. But we also were really interested in how much energy does a parent have to put out to bring fish for the chicks. So if you haven't spent half your life watching murre colonies, they're pretty fun to watch the parents come in and feed the chicks, because parents always capture only one fish at a time. So puffins, for instance, might capture 5, 10, 15, even almost 20 fish and stack them in their bill. And that's because they have a rigid backward pointing tongue and they can use it to keep all the fish in place, which is very cool. But murres, they only fish one fish at a time, and they always bring it back head in and tail out. So I became an expert at identifying fish butts. And in fact, that was my first key, was a key to fish butts. So we could teach graduate students and our summer employees how to identify fish. So we're interested here in this diagram using Tatoosh data on how big those fish are, and basically what the volume of the fish is. And we're looking at anomaly plots, so 0 is the average. And on the y-axis, we're looking at provisioning rate, which is basically how often parents are coming back and giving the chick fish. And what you can see here is that there are 3 roughly sets of years. Really bad years, the years in red or orange, in which the energy per fish is pretty low and the provisioning rate is pretty low. These are years in which we see a lot of dead adults on the beach during the breeding season because there's just no fish out there. These are years in which the murres might, in fact, abandon breeding because it's just too stressful to them. Contrast that with good years, which are in the upper right-hand corner. These are years in which the fish are pretty fat and sassy, so the energy per fish is high, and also the provisioning rate can be high as well. So the chicks are getting so much to eat, they literally cannot accept any more fish from the parents. And then there are compensation years in which the provisioning rate is really high, but the energy per fish is low. Obviously, the parents are trying to make up that difference, right? And get their kid enough fish to fledge the kid for the years. So they are accommodating. So what this graph shows over years and years of looking at a colony is that the adult murres, who live a long time, about 20 years on average, or maybe actually extended, they can accommodate shifts in climate. So the shifts in climate from year to year are going to change the production of the system. Production of the system is going to change the availability of the forage fish. And here are the murre parents trying to make up for it. And they can do it in some years, but not in all years. So even back then, without really thinking about it, we were already looking at the effects of climate on murres. Most of what I want to talk about today is not colony work, but actually is work on dead birds, which is where my real heart is now. I'm a dead bird lady, which I really never thought I would say, but there it is. Years and years after I pass on and everybody stops reading my scientific publications as if they read them now, my largest selling publications will be my field guide to dead birds, of which I have 3 different ones. And some of them are in their 5th printing. So I have tens of thousands of field guides to dead birds out there in circulation. And so I'm the dead bird lady. So I want to get you to think about this question because we're going to return to it at the end of the talk. Can you use citizen science in the toolbox of science? And I asked this question last night more for a public audience, but for a science audience, you know, this is, for me, an honest question. So there are lots of different methodologies that we use in science to collect our data. And of course, we want it to be correct and we want it to be precise to whatever it is, whatever phenomena it is that we're trying to figure out. And so an open question for me is, can you involve the public in the work that you do in ways that they're collecting information? And really, 2 good things to think about are that a citizen science program should at least ideally be authentic science. That is, we don't pre-know the answer. We are actually getting people to be involved in our science team and collect information that we can't get any other way. And I'm putting forward to you that citizen science is actually quite good at collecting environmental data, including biodiversity data, in ways that we just really can't do any other way. But the downside of it is that citizen science is free choice learning. And by that, what I mean is that the individual has a free choice to stay and continue participating in the program, or to go because it's boring, or they're not getting any feedback, or because the feedback they're getting is not very good or very positive. And there are all sorts of indications that the group of people that are involved in any one citizen science program reflects all of those things, as well as their lived experience. For instance, there was a really, really wonderful study done in the 20-teens looking at bird programs writ large, both in the US and the UK, and just looking at gender distribution and just to gender, male or female. And they showed that for programs that had a really strong community component to membership, so it wasn't just about going and collecting data, it was about coming together, sharing stories, really getting to know your neighbors, about 80% female. And the more competitive it got and hierarchical, so there are beginners and there are masters and there are uber birders and there are those that can check the veracity of others, the more you got towards competition, up to 92% male. So that's a pretty wide swing, 92% male to 80% female, and that's just gender. So when we think about citizen science programs, although there may be a citizen science program for everybody in the world, everybody is not equally invited to the same program. So I want you to keep that in mind as well. So I'm going to talk to you for the rest of today about the Coastal Observation and Seabird Survey Team, or COAST for short. And in COAST, we're doing standardized search. So the same way, everybody does it the same way. They repeat monthly or as often as they can. Things are effort controlled, so we know how many people are out there. There's a set start point and a set turnaround point. It's a single search pass of the beach. And they're collecting basic data that allow them to deduce what species it is, and we check them on that. So they're doing beach cast birds, dead birds, not live birds. And relevant back to my title, let me tell you that common murres are our #1 species, coming in at about 30% of all of the carcasses that coasters have found. So here's a little bit of the coast conceptual model, really, really high level. So climate, of course, is producing weather, but climate also has a direct impact on the physics of the system. The physics is impacting the biology all the way on up through trophic transfer. So, right, physics is bottom up, it's coming, it's changing production, that's changing a set of trophic levels, which may or may not whack off part of the top of the trophic pyramid. But physics also has a lot of abiotic forcing that is impacting upper trophics. So for instance, climate warming may in fact hit a species' thermal tolerance. That's an impact of a direct effect that's not going through trophic transfer. So there are 3 things to think about with respect to the coast model. And that is that the last thing that a bird does before it becomes moribund is it has chosen where to be. Right? So it's made an ecological decision about where to go, which may be constrained by breeding, but it's probably also constrained by food. And then mortality is going to shift as a function of location, right? So just broadly speaking, and where birds are is going to shift the probability that they'll be deposited on a beach. Okay, so just a few things about COAST. It's about 25 years old. Right now, we maintain about 400 sites from just south of Mendocino in Northern California on up to Uktiavik, the former Barrow, up above the Arctic Circle, and over to the Commander Islands in Russia. We work in about 90 different coastal communities, and right now have about 700 people in the program. And over all of those years, we found about 100,000 carcasses. So that's a pretty big data set for us to say something not only about common species like common murres, but also system-wide. And here is the big thing we do. I showed these graphs last night, so you can check me on it and see if I say the same thing if you were here yesterday. This is what I call the natural history of dead birds, because in it, we get a lot of clues to what's going on with the birds. So how can you interpret this graphic? On the y-axis is how many carcasses you might expect to find in a lineal kilometer of beach. And the data that I'm showing you comes from Northern Oregon. So all of the beaches that we have in Northern Oregon, and the black line, which is the long-term average, is all of the beaches over all of the year. So every time new data comes into the coast data set, ostensibly these graphs change just a tiny little bit. But with 100,000 carcasses, you can imagine they don't change very much anymore. And you can see that there is a lot going on here during the late summer, fall, and into early winter. You can see that there are more carcasses per kilometer, goes down at the end of winter into the spring and early summer. The gray bars are what happened in any given year. So here I'm showing you April 2016 through May 2017. And in case you're wondering about what makes the hills and valleys here, hills are created by predominantly 2 normal things: post-breeding mortality, that's the first peak, exhausted parents and inept chicks, right? Because juvenile mortality is always higher than adult mortality. And the second peak is winter kill. So decreasing temperature, increasing storms, increasing exposure is in fact the #1 killer of birds worldwide, whether they're marine or terrestrial. And so you can see that in this particular year, 2016, 2017, it was a pretty normal year. That is to say that the gray bars follow the long-term average, that black line with the yellow wash. But that is not always the case. I can take that same long-term average and I can print it back across the previous year also in Northern Oregon, and it looks like this. So that curve has flattened out quite a bit, and that's because I've had to stretch the y-axis. The previous graphic, that was at 6 carcasses per kilometer. Here it's up at 14. So that's a little more than twice. Remember that coasters go out once a month, but each team goes out at a separate time. And so we can come up with a broader picture than just a single month snapshot. So when we produce these graphics, which you can do yourself on the Coast website if you like to be a data wonk and interact with Coast graphics, you can see that that wow out of post-breeding mortality equals out to about 3,000 carcasses per Northern Oregon for every day. So that's a lot of carcasses. This is pretty much a not normal year. But I can go back a year prior to that, 2014, 2015, and you can see now a super not normal year. So there, the long-term average is almost a flat line, right? Because the wow out, which is now in red on the Coast website, whenever there's an event like that that is 5 times above whatever max carcasses per kilometer we've seen, we make it red, so you can see it, as if you couldn't see those bars way up there before. But just to say, if you do the simple math, that's about 8,000 carcasses per day over Northern Oregon. That lasted for about 2 months. So that's a lot of birds. That's a really not normal year. So what do we do with those basic count data? I'm going to kind of walk you through this. Is there a pointer?
Use your pointer.
Ah, okay.
Just your mouse pointer.
Cool. All right. Follow along with the bouncing mouse pointer. So Coast data are carcass count data. And so when we are thinking about data, we're getting data from beach bird surveys, which I've just talked to you about. We also have done daily survey studies where some amazing volunteers have gone out sequential days, up to 10 days sequentially every month for up to 2 years, right, in place. And that gives us a really good idea of what comes to shore. We use the daily survey data to populate a capture mark recapture, or a CMR model. And that tells us a lot about what is found versus unfound on the beach. So we can get detection probabilities and persistence probabilities using our CMR model that's populated with a daily survey rate, and essentially fill in the blanks of what we aren't seeing either in time because there wasn't a survey that day, or in space because that kilometer is not surveyed. We are actually trying to get back to total deposition. That is, how many birds had to wash ashore and for us to get the carcass counts that we did get. So we're a little bit using the CMR model for that, but we're also bounding things in space and time. So how many kilometers are depositionable? For instance, river mouths don't count. And over what period of time. What we really want to get back to is required mortality. That is, how many birds had to die at sea in order for us to get the estimated deposition that we got in order for us to get the carcass count. So what we actually want to do is run the model backwards in time. And in order to do that, we need a different set of models, and in this case, simulations. First of all, it's great if we have at-sea distribution data. That is actual, in the moment, data on where the birds what we're interested in for a mass mortality event, where they are. We often don't have that, so we have to make simplifying assumptions, for instance, a uniform distribution. But we use an ocean circulation simulation, which I'll show you a little bit of, and that's the best thing to have. Failing that, we use wind field data. We also work into that a sink rate model. Once you die, you're not going to float forever. You're not a piece of plastic, right? So those carcasses will sink over time. And there are lots of people who have done some amazing sink rate studies, and we use those. All of that goes into trying to figure out the difference between potential deposition and loss at sea. So depending on where a carcass or where a bird became a carcass or became a moribund bird. There are all sorts of factors that may get it to shore or not. So this is the sort of overall graphic of what we do. So let me dive in a little bit to the ocean circulation model because it's fun. So this is a long-term collaboration with Parker McCready, who's a physical oceanographer, and he has created an ocean circulation model that's very, very fine scale called Live Ocean. And he actually has another linked model for the Salish Sea. And it tells you in 4D, so surface area and depth over time, where a particle of ocean is likely to go. It also has a lot of other stuff about salinity and temperature, a bunch of other stuff. And it is forced by currents and tides and wind fields. And what we can do in the surface version of Live Ocean is we can put in a set of particles that are the size of the carcasses that we're interested in, and we can assign a windage to those carcasses. That is, we can know how far above the water they're going to float, at least to begin with, and treat them thusly. Of course, the farther above the surface they float, the more they're going to move as a function of wind versus just current. Okay, so I'm going to show you two simulations of Live Ocean. So each triangle here is a carcass. And I'm going to start one on the 19th of December and one on the 22nd of December. These are from 2013. And we're going to run these for 7 days. And you can see the little curlicues that are happening. Those are tidal cycles as the whole ocean is sort of moving in and out of the space. When the triangles get within a kilometer of shore, we stick them to the beach. So you can see that when they hit the shore, they land there. And we're just finishing on the 22nd of December. So one thing to note, a bird that died on the 19th or became moribund on the 19th of December has a far greater chance of reaching the shore than a bird that died just 3 days later, right? So there are small scale events in the coastal ocean here, 3 to 5 day events that may really, really change the chance that a carcass gets to shore. So upwelling, downwelling shifts in that small time scale. Of course, we have upwelling and downwelling that's changing over a seasonal cycle, right? Upwelling during the spring, summer, downwelling during the fall, winter. But if you actually look day to day to day, there are these event scale reversals that happen all the time. It's just that the propensity is towards upwelling in the spring, summer, and towards downwelling in the fall, winter. So we use this live ocean data and we run simulations for every day going forward. And these simulations have been done without a sink rate model. So we get to where the birds would have been, and then we apply a sink rate model across all of the carcasses. And that tells us how many birds would have gotten to shore, what the probability is. And we can build up a probability distribution over the entire time that a mass mortality event is really happening. And you can see here for different squares of the model, if I let the carcasses persist float for 3 days versus 5 days versus 7 days, I can look at what the chance is if it started in a particular square that it would get to shore. And this is a no-brainer. The closer the square is to shore, the higher the probability. So it's yellow towards red in the direction of the shore. But notice all of the gray. Gray means that no matter what simulation we ran, if carcasses started in those places in the ocean, they never, ever, ever get to shore. And that solid line is the shelf. Okay. So I'm going to tell you 2 stories that are incorporating the live ocean model and sink rate model and our capture mark, recapture model and all sorts of models. And I promise I'm going to show you no math. I thought about it and I decided, who needs math? So you'll just have to believe me or you'll have to call up Tim. And so one is a data rich story where we have all that stuff. And then I'm going to tell you some data poor stories. So our data rich story is a story about Cassin's auklet. This is a small bird about the size of my fist. It is a burrow nesting bird. It's also an Alcid. It's related to the murres. So it's a little football, like a Nerf football with wings. but it's eating lower on the food chain. So this is primarily a zooplanktivore, although it will also eat larval fish. The auklets breed and are distributed over the entire west coast, but the center of their breeding population is Triangle Island, which is that big star at the northwest tip of Vancouver Island. Okay, so these are the data that I showed you before. There's that 2-month wow out. And now you can see that wow out was, in fact, almost all Cassin's auklets. So this is one really interesting and quite particular thing about mass mortality events that we see, which is that when we have mass mortality events, it is not that all birds come or wash in more frequently. It's that we have a focus in one part of the seabird community. And it's usually either planktivores or it's piscivores. We never see both at the same time. Okay, so this story is about Cassins. And this story actually has a whole bunch of different data sets attached to it. You can see that sort of purple box labeled LO, that's live ocean. That's the domain of the model. You can see that it's all of the coast of Washington, goes down into Oregon, but it also goes up into Vancouver Island, but not all of Vancouver Island. So within that blue box, it's great, right? We've got a great fine scale ocean circulation model. We also have lots and lots and lots of beach bird data. Like that's about as much as we want. We also have our daily survey data from that same box. So we're serving up like the highest and best quality data that we have. South of that, we have lots of beach bird data, but we don't have an ocean circulation model. North of that, we have an ocean circulation model, but we don't have any beach bird data. And way north of that, we have neither. However, the Cassins didn't pay attention, right? So they're dying over that entire space. So if we want to responsibly try and figure out what's the bounding box for required mortality, we can't just do the thing that scientists do, which is put blinders on and say, I'm only going to look at the data-rich case, right? And I'm just going to tell you that because I can give you a bounding estimate, and I'm going to just pretend that other space didn't exist. We have to pretend everything exists. So lots of work that we've done on this, but I'm just showing you part of it. So here are the wind fields that are forcing the live ocean model. And the middle, let me just put that up here. So this is what's going on at the ocean surface. This middle graphic here is what the circulation and the sink rate models suggest. So the black line is the beaching probability, and the red line is the daily arrival rate. Daily arrival rate is going to be slightly spikier. The beaching probability is a bit smooth, and that's because carcasses have a variable sink rate. So on every day, carcasses are coming in that died that day and the day before and the day before. And what is the panel C? That's what was actually observed. So a few things that I want you to note about this. One is that what is actually observed is lagged about 2-3 weeks from where we predicted carcasses would come in, where the beaching probability was high. Why? Coasters aren't out the very day that something happens, right? They may be out a week, 2 weeks, a month later. So they're collecting information sometimes about carcasses that just died and washed in, and we have ways of knowing that, but they are showing us a window to the slight past. So our expectation is that there actually should be a little bit of a lag, which there is. So one of the things that we can answer here is yes, the live ocean circulation models suggest to us when it was possible, if there were moribund carcasses available, they would have washed in, and they match reality. So two things, right? The physics has to line up in the right direction. That's what I'm showing you here. But so does the ecology. That is, the birds have to be in the area and be moribund. So where were they? The map here is showing data from many years of satellite tagging of Cassins geolocation tagging on Cassins from Triangle Island and from Frederick Island, which is at the northwest tip of Haida Gwaii, the Queen Charlotte Islands. And what you can see is they smear all up and down. They stay in the BC area. Some actually go up towards the Gulf of Alaska, and they also smear down the coast. The panel on the other side is NOAA triennial survey data. So happening every 3 years. It's a fish survey, but there are also bird counters aboard. And in fact, the bird count surveys are run out of here. And they were actually provided to us by Lisa Balance and her students. And I just want to point your attention to the 2014 line. That's that blue line. Really, really different from the other years. So this is one of the cautions about trying to figure out where the birds are. What's the at-sea distribution? Because if I had used the average, I would have gotten the answer wrong, right? In that year, in 2014, they were actually super snugged up to the coast. And I'm going to explain that in a minute. So we actually used both a uniform distribution in our simulations, but we also used the NOAA triennial survey data. And here's a sensitivity of the whole model for deposition and for required mortality. And you can see that over all of the things that we put into the models, persistence actually is pretty sensitive. So if we shift persistence just a little bit, we're getting a pretty good shift in deposition rate. That goes down a little bit when you get to mortality. That's the longer graph. Mortality, of course, is also a big function of at-sea distribution. So these parameters are really, really pretty sensitive. And so it turns out that it's pretty important to know what the at-sea distribution is. And that's actually a really hard thing, it turns out, to know. Okay. So we did this and came up with an estimate in the live ocean range. But what happens when we go to the data poor place? So the data poor place is Vancouver Island all the way on up to Haida Gwaii. And just more or less, here's what we did. So the first thing that we did is we extended the deposition we calculated in northern Washington. We have a lot of confidence in those data. And so we pulled that north onto Vancouver Island. And we did that because we assumed that extension when we did our simulation models from live ocean. Remember that the live ocean domain goes up to half of Vancouver Island. And we could see, and you can see that in panel D, that, that there's a fairly high deposition probability, even from carcasses that originate in Washington waters. And then we used survey data and or anecdotal data from Vancouver Island and from Haida Gwaii, that's the Queen Charlotte Islands, to say, yes, there were carcasses here, or no, there were not carcasses here at all. So we have no real information on encounter rate at all. We're just making an assumption, and we're taking that assumption and we're spreading it north over time, and we're doing it in a uniform way, which is why there's that sort of shelf that you see at number 1. And we're rating that whole thing according to the NOAA at-sea distribution. Remember that the birds were snugged up really close to the shore. So I'm going to tell you what the answer is to how many birds died, but first I want to segue a little bit into why. So we also, for this particular study, could ask the question why, and that's because we had a lot of collaborators that were collecting a lot of information about how productive is the ocean, how productive are the birds on the colony on Triangle Island. We could get storminess from significant wave height, and from right here, from Bill Peterson's amazing Newport line data of copepod distribution, we could get food. So we actually constructed a whole bunch of GAMs and GLIMs, these are GLIMs, looking at the impacts of various things on encounter rate. Each one of those circles is a year. The top panels are with 2014 included. 2014 was that big wow out year, and the bottom panels are 2014 excluded. And the models suggest to us that bad food, that is the proportion of southern copepods in the system, really set the stage for encounter rate, as does the amount of bad habitat. That is, for the near shore ocean, the proportion of the surface waters that are warmer than long-term normal. Those two things really set the stage for encounter rate. Not a big surprise. But even when you exclude 2014, which is that number that's pulling up the whole relationship, we still get a relationship with copepods with bad food, but instead we get a relationship with storminess. So in a normal year, storminess actually does play a role in how many birds are going to die, at least how many castings are going to die. So here's what kind of we saw. This top panel here is Newport line data, and the bottom panel is data at Trinidad Head. The black line is the sea surface temperature anomaly. And you can see that standard, right? When during the upwelling season, the temperature goes down. When upwelling stops, the temperature goes back up. Again, the colored lines are the copepod anomalies on the top and the krill size anomalies on the bottom. So basically, we had a sudden loss of cold water when upwelling ceased at the end of the season, and that actually advected in new water masses. And what we saw was a very sudden shift towards smaller copepods and towards smaller krill. And this changed the amount of energy that Cassin's auklets had to expend until they got to a negative ROI. That is, they're expending more energy to get the food than they are getting from the food. And that caused mass starvation. And all of those birds were snugged into the coastal ocean. And why? Because that was the only place that the blob, the marine heat wave, had not managed to invade because of seasonal upwelling, which of course dissipated at the end of the season. Too bad for them. Our models suggest that about 400,000 Cassins lost their lives. That's a bounded estimate there, 8 to 10% of the world's population. That's a pretty big number, but it is also the same as a good productive year from Triangle Island alone. So begs the question if this mortality was additive or compensatory, right? So if all of these birds were killed off and thus the juvenile survival rate was higher, we wouldn't see a long-term population impact. Versus if it wasn't, and we have normal high juvenile mortality plus adult mortality, it would have been a longer-term impact. Okay, I want to tell one more story. Actually, I'm lying. I might want to tell 2 more stories, but I'm going to tell you about common murres. Common murres, there are many more of them than there are Cassin's auklets. Common murres are also both in the Pacific and the Atlantic. There are arguably 20, 25 million of those birds. They're slightly larger, so they're piscivorous. They actually are the size of an actual football. In the year following the Cassins die-off, in that next winter, that's when we saw a murre die-off. And this die-off was wider, broader in distribution than the Cassins die-off. The Cassins die-off went up through BC, but the murre die-off, we picked it up in the lower 48, but it was really centered in the Gulf of Alaska and actually on up into the eastern Bering Sea. And you can see here, here we are showing coast data and how far above normal we're seeing mortality. So in Northern California, was about 5x above normal. In Oregon and Washington, for the 2 months of the event in the lower 48, it was about 10x above normal. In Alaska, it was 1,000 times above normal for 9 months running. So even though there were 2 months that were large, there was a really long-term signal that started actually at the beginning of the breeding season in May and persisted well into the winter. So those birds were definitely in trouble. Now, we don't have a live ocean model. We don't have any kind of data that allow us to get to a well-bounded required mortality estimate in Alaska. So we had to stop at just saying how far above normal it is. But we also made a conjecture about what happened, which is, again, through trophic transfer, the physics are changing the ecology of the system, changing prey size and availability. We know that happened because other people have recorded that. Other people have also gone on to look at the metabolism of the large fish, suggesting that that increased. They became basically more voracious predators. So more voracious predators and a change in prey size means that murres were basically outcompeted by large predatory fish and starved to death. Okay, we have some suggestion though from colony data, and this is a paper that we just this week, yay, got accepted in Science, looking at colony numbers on monitored colonies prior to the heat wave, during the heat wave, and after the heat wave. And what we can see from these data is there was a 50 to 75% loss across the monitored colonies. And if that is not bad enough, that once that loss happened, you would expect then the populations would recover, right? Because there's the system goes back to some sense of what it was before. So there's a lot more food, competition should go down, and there's a lot of breeding sites available. However, 8 years post heat wave, we haven't seen that recovery of the Alaska murres yet. So this is the first signal that we have that there seems to be a long-standing impact or effect of a sudden shift in climate. Okay. One more quick story. This is my my most data poor story. And it's about tufted puffins. And it happened yet in the next year. So this is towards the end of the heat wave. This is a story about St. Paul in the Pribilof Islands, between St. Paul and St. George in the Pribilofs. We think jazz hands are about 7,000 or were 7,000 puffins breeding on the island. This is a story that's done in conjunction with our partners on island, Bearing Watch, and in the Ecosystem Conservation Office of the Aleut community of St. Paul. And without the permission of the Aleut community and our partners in ECO, we could not work there. So here's St. Paul, a little island out towards the shelf in the middle of nowhere. And here's a photograph that we got from the ECO office. They do coast as part of their regular surveys. It's a beautiful photograph, I think. It's really well lined up. We can see what everything is. They labeled it for us, and we got many, many more of these photographs. This was day one of what they saw, mostly tufted puffins. We can actually look at the encounter rate and also the effort. So we saw a spike in encounter rate, but we also did have a spike in effort. So when we are looking at estimating what went on there, we have to take that into account. Over the entire mass mortality event, or MME, in the winter of 16-17, we only found about 357 carcasses. So that's a small number relative to the other stories that I've been telling you, but it's a small island out in the middle of nowhere, right? It is not the entire West Coast. It's one island. 3/4 of those carcasses were adult tufted puffins. There were also juvenile tufted puffins. That's that orange slice. So the encounter rates that we saw were 60 to 70 times normal. We also have two other really interesting pieces of evidence, and that is that of the carcasses that came in, about 95% of them were in molt. Tufted puffins go through a catastrophic molt, which means they lose all their flight feathers at once. They are sitting ducks. They cannot fly. So that what they need to do from a natural history point of view, finish breeding, migrate to where they're going to spend the winter in a food-rich place, get there before they lose their feathers, right? That's their assignment. Didn't happen in this year. The other really interesting piece of information that we have is 75% of the carcasses are completely intact, which is interesting because both of the Pribs have endemic Arctic foxes, and they persist in the fall and winter on what the tide brings in. I personally have wrestled with an Arctic fox over a northern fulmar carcass. Although I'm here to tell you the story, the fox won. So 75% intact means that we have maxed out the scavenger population on the island. We couldn't use a live ocean model, but we could use the NCEP wind reanalysis data. And a really interesting phenomena happened. We have long-term monitoring beaches on St. Paul, St. George, which is about 35 kilometers away. We used to have beaches there. We do not any longer, but the community goes out regularly. So we have anecdotal data from St. George. This event happened only on St. Paul, not on St. George. So what we could do is we could set up simulations using the wind, seeing how far away from St. Paul as the center of the universe, that's the bullseye in those graphics above, a carcass had to be to get to St. George or St. Paul red, but not St. George blue. Okay. And in fact, this is another way of looking at that proportional deposition rates. The solid lines are St. Paul, the dotted lines are St. George. And what they suggest is that there was about a 20-kilometer maximum window or range around St. Paul that the carcasses were in, because outside of that, we would have had them reported on St. George as well. And that set up a window that we could look at required mortality in. So that was a very data poor way of going about it. And what we discovered is that our estimates are broader, but up to above 7,000 carcasses. Remember, I told you that there were only 7,000 puffins that bred there. So we know that it didn't wipe out the population of puffins in the Pribilofs. We actually think what happened is that puffins were coming from many different colonies and moving out to the edge of the shelf. They were delaying move because there was that was a very food stressed year, and they delayed too much and they ended up on the shelf. So we could bound this on St. Paul, but we have no idea, in fact, how many carcasses died in total. So are there larger lessons that we can draw? One thing to think about is the story with warm water. So these are ribbon plots starting from California in the south, going to the Chukchi in the north over time. And here you can see the Northeast Pacific marine heat wave, or the blob. That's when the ocean got stuck on warm at the end of 2013, at the beginning of 2014, right? But there are two other phenomena that I want to point out to you here. One is the loss of Arctic sea ice. So at the same time that the marine heat wave was hitting, we were having a long-term shift from a cold stanza to a warm stanza in the Arctic. And this really, really changed how much sea ice we saw in the Chukchi and down into the Bering. So from the north, we were also getting a warming impact, and we were getting it from the marine heat wave. And then on the backside of the marine heat wave, there was an El Nino, which extended the blob in the southern part of its range. And that brings up the question, marine heat wave. What is a marine heat wave? Everybody's getting into the marine heat wave business these days. So now people are saying, 3 to 5 contiguous days of sea surface temperatures at 90% or even 99% of the long-term climatology counts. 3 days of hot weather, a bird's going to go, whatever. So a bird is really looking at a longer term. So we're looking here from a bird point of view as a marine heat wave as a discrete and prolonged anomalously warm water event. So I showed you this graphic in the beginning, and I want to return to it, right? If we think about a 3 to 5 day anomaly, that's down in the days to weeks. From a seabird's point of view, we are really not anywhere. But if we think about a marine heat wave as one plus standard deviation in anomaly for 2 sequential seasons, spring, summer, summer, fall, fall, winter, or more, then we're going to have an impact. Remember, because I've put ecosystem forcing at the seasonal level, and ecosystem shift up at years. So when we see upper trophic UT mass mortality events, it's going to be in between those. And so really think about a marine heat wave as that long-term, prolonged, and also large-scale over space forcing event. Okay, for this analysis, we actually used coast data, but we actually added 3 more beach bird programs, BCBBS in British Columbia, and then 2 in California. So 4 citizen science programs, 30 years, and 90,000 surveys. And what I'm going to show you here is a track of time of mass mortality events that we saw, where each one is a circle, and they're all keyed to each other. So bigger circles are more mortality. And you can see here that before the marine heat wave happened, when the ocean got stuck on warm, there were mass mortality events of many different species, and the circles are less frequent and they are smaller. But once the marine heat wave hit, we saw more. So mass mortality events went up in frequency and they got larger. And they also honed in taxonomically on 2 families, the Alcids, I've told you those stories, and the Procellareids, the tube noses, including northern fulmars and shearwaters. Okay, so this is the way that we are looking at that. On the x-axis, you're looking at annual sea surface temperature anomaly, right? So long-term normal is set at 0. Less than that is cold, more than that is warm. And we're asking a series of questions. Each one of these dots is a mass mortality event. The ones in black are the big ones. The ones in gray are the noise. So I want you to look at the category 2 plus the black. Okay, and we can ask a few questions. Is the body count relevant to sea surface temperature? And the answer is really no, not so much. So carcass encounter rate is not really forced by sea surface temperature anomaly. But how fast are they washing ashore? Sharp increase. Oh, I'm sorry. Body count, not so much. Encounter rate, carcasses per kilometer, is an increase. Also, how long does the event last, or the duration? Mass mortality events definitely last longer when the water is warmer. And there is a mild effect of space. They occur over larger space. We can actually take those three things, encounter rate and duration and geographic extent, and we can put them into a single dimensionless variable magnitude and look at that. And we see a serious step function, which is basically a 5-fold increase between 0, normal temperature, and +1 in anomaly, which literally translates into millions of birds dying. Okay. The last thing that I want to show you is that the long-term impact. One thing that we noticed when we did this analysis is when there are mass mortality events and when there's a marine heat wave, there are 3 different marine heat waves that I'm showing you in these panels. The dashed line that goes all the way down through the panels is the start of the marine heat wave. 3 separate events. Okay. And what we can see is that when we get a marine heat wave, that is more than 1 degree lasting for 2+ seasons, we get a mass mortality event of seabirds within the first 6 months. Everywhere. And then we get another one the next year, right? So this appears to be a consistent pattern when we have persistent marine heat waves. And then very, very interestingly, the line goes blue. We have another 10 to 16 months of lower than normal carcasses. So there appears to be a recovery period, at least in the seabird community. One interpretation of this is that there's overkill during the mass mortality events. And so competition is relaxed when the production in the system goes back, and that elevates survival, depresses mortality, and so we're seeing less. But what we know is that when we get a marine heat wave, it's going to take about 3 years for the system to work its way through mortality events and a recovery, right? And so the question is, how frequent are marine heat waves? And are they going to become as frequent as 3 years or 3 to 5 years, giving basically the seabirds not enough time to recover? Okay, I think, yes, I want to stop there. So asking the question, will common murres become uncommon? When I was doing ecology and work on colonies, I would have said, yeah, absolutely, because of what we're doing to them. Then when we started to work on effects of climate, I would have said in the lower 48, no, not so much. They're actually still pretty common here. But in Alaska, we've seen the first evidence of a long-term and persistent shift in a very large population because provoked by climate. So the final thing that I want to say is marine heat waves give us a window into the future, right? And when we see mass mortality events, what they are responding to is the sudden shift in the climate over a very large amount of space, right? So we don't expect a future of mass mortality events. We expect a future of lower production and inability for some species to exist. So what we're seeing in the present with mass mortality events is this window onto energy flows through the system and who's going to persist in the system and who's going to lose. And that may be murres. Thanks for listening.
Thank you so very, very much. I know that we're at our hour, so if folks need to go trick-or-treating, go ahead and exit out the back for us. But if you have questions, stick around, and we're going to bounce back and forth between questions online and questions in the room. Roseanne, it looks like you don't have a question yet. So folks online, if you have any questions, go ahead and type them into the chat box, and we'll get to them. For folks in the room, just raise your hand. I'll get you a mic. Any questions? You can't let Dr. Parrish off so easy. Come on now. All right. You're going to make me run.
So on the graphs that you showed with the puffin mortality spikes, there was a really big mortality event in 2008, I think. What was that about?
Wow, test my recall. I believe that was a shearwater event. Although I wouldn't swear to it. So shearwaters are really interesting species. They congregate on the water in the open ocean in huge numbers and very, very, very dense. And they are a species that appears to die in fairly large numbers in open ocean situations. So it's actually kind of interesting and unusual that we see shearwaters wash up on the beach because they don't come close to land. But the pribs are way out there. And so when we have a shearwater die-off, we do tend to see them on the prib. So if you look at the whole distribution of what carcasses are found on the pribilophs, most of them are procellareids, and they're going to be two species of shearwaters and then northern fulmars. So those are the usual species we see. Great question.
Nice. I see a question online. Go ahead, Roseanne.
Are there data sets for forage fish community shifts that coincide with some of these MMEs?
Really good question. I wish that I could say yes. I wish that I could say absolutely. Marine scientists have been able to convince those with money, the federal government, I don't know, to do forage fish surveys everywhere. But Waldo will tell you how many forage fish surveys we have. Not a lot. So some places, yes. For instance, in the Gulf of Alaska, during the murre mass mortality event, we actually had some forage fish surveys in some places. Gulf of Alaska is a big place, suggesting that there were shifts in relative species abundance and also in size of fish. But often we can't make that step. I would love to say that we could. Here's a plug for forage fish surveys everywhere.
It looks like Roseanne is a follow-up. Hang on a second. Okay, hang on just a second.
Thanks, Julia. As a birder, I not infrequently get asked when I'm on the beach, somebody will be like, oh, what's this dead bird? Is that unusual? Or what kind of bird is this? So I was wondering how often, like during these mass mortality events, does that translate into action for people on the beach? Does that lead to a spike in recruitment for coast?
It's actually the other way around. So I know this sounds perverse, but there's a lot of joy in going to the beach and finding a dead bird. I know it sounds weird, but that's because you have a set of tools, you get to collect all the information, you get to use the guide. It's not a guessing guide, it's a dichotomous key. Many people have that aha moment, especially if it's a species they haven't found yet, or a truly rare species. And in patiently examining the carcass, you can actually learn a lot about the natural history, how that bird made its living. So there's a lot to be learned and gained by that patient observation of natural history. However, when you go to the beach and you cannot even walk a meter before you see another dead bird and another dead bird, you switch from I'm in coast mode and I'm being the natural history observer to, oh my God, I'm being overwhelmed by birds. Also, when a mass mortality event is happening, moribund carcasses will be coming in. These are birds that cannot fly, cannot swim. They're rolling, they're still alive. They're rolling around in the waves. It's heartbreaking. And people want to try and rescue. Those birds they're gone. They've lost so much weight, they won't recover. So we actually lose more people from coast during mass mortality events because they just can't put that sort of coast visor down and say, I'm going to tough through it. We've actually done, I have a whole social science side of coast that we run, asking questions about learning and about identity and belonging. And people say they just get overwhelmed. So yeah, it seems like it should be the other way around, but we do get a lot of calls for action. And what I can say is coast data, they go into science, but they also go into natural resource management. So coast data, for instance, are delivered to both of the fishery management councils, North Pacific and the Alaska Fishery Management Council, as one measure of ecosystem health. I have done testimony in front of the Washington state legislature on multiple occasions and twice in DC. And that's because staffers called me up to do that. And that's because local coasters called them up and said, this is really important. This is going on. You need to hear about this and you guys need to do something about it. So my voice as a scientist gets way amplified because I have hundreds and hundreds of people that live in that place and know it and know what's going on and just, put their foot down and say, this has got to change. So we do get those calls to action. Unfortunately, my ability to turn the dial down on climate change, I can't do that, but I can make people aware. And my sense is awareness really can make a difference if you connect it directly to things that people care about in the community. Good question, thanks.
I think that's a great wrap up, but I know that Roseanne has a question that is burning, so I'm going to hand it back to Roseanne. Go ahead.
Okay, if common murres are particularly susceptible to heat waves, are there species that are exceptionally resilient?
Here are species that we haven't seen connected to heat waves in that 30-year data set. None of the larids, the gulls, they seem to make it through, and they are fairly nearshore birds, so we would see them. But they are pretty resilient. They have lots and lots and lots of different food sources. Also, they have a slightly less all eggs in one basket. One egg in a basket. There, how's that? That's what a murre would be. So we don't see larids. We don't see phallocrichorids, the cormorants, also near shore, also a pretty diverse diet. We will occasionally see loons and grebes, but only during a very narrow window when they're molting. They're also catastrophic molters. They'll also lose all their flight feathers and be sitting ducks. And we don't see marine waterfowl, of which we have many, many hundreds of thousands. Scoters, for instance, we don't see at all. So there do appear to be winners in the system, mainly near shore. And then within the procellareids, the tube noses, although we see lots of fulmars and shearwaters, we don't see albatross and we don't see storm petrels. So real differences. And I wish that I could tell you why, but I'm not that smart.
All right, we're going to wrap it up there. Everyone, go ahead and give a round of applause for Julia. Thanks. Thank you so much for being here. Everyone, drive safe tonight and enjoy your Halloween festivities. We'll see you next week.